Goedenavond allemaal. Allereerst gefeliciteerd Esther en het bestuur met het geweldig rapport. I'm switching to English because our speaker don't understand Dutch yet. And we have also some audience, not only from the Netherlands, also from abroad. So NVVS Go International. Welcome to this marvelous webinar. I'm sure that it will be very interesting. And before we start, some information about, I don't know why it not moved, but now probably we'll do it, about cannabis. In the Netherlands in general, we know it, but cannabis is one of the most used drugs worldwide, and it's contain actually group of psychoactive plants. Not everyone knows that there is difference between the plants. And we have the sedativa, which is mainly stimulants, the indica, which mainly about relaxation and have less central effect, and ruderalis, which use a lot of time in the medical cannabis. We are all aware that there are about 120 compounds in the cannabis plant, and from only 2 of them we are more or less understanding, and this is the CBD, which is a non-intoxicating and non-aphoretic, less central effects, and the THC, which have mainly psychoactive compounds. But time changing and the use of cannabis as well, which was in the past and a drug with a lot of stigma, became used in the 60s for a lot of social reasons. And in the last 20 years, we saw increasing use for medical reasons, and nowadays even for quality of life. And just to give all of you an example, how many indication nowadays are used, there is a use off-label yet, of cannabis, like ADHD and Alzheimer, in PTSS, in chronic pain and rheumatoid arthritis, and so on and so on. And as you can see from this slide, you see that there is different compounds and different doses of the CBD and the THC. So it became more and more complicated in use. And what about cannabis and sex? It became also very interesting, and that was one of the reasons to choose for this webinar. And Jim held during the ESSM in Prague a marvelous combat boxing fight with Michal Lev from Poland about the pro and con of cannabis, which he showed a lot of knowledge about the positive effects concerning sexual benefits. But we also know that there are some association with the erectile dysfunction or reduction of testosterone. And we will hear much more about it in a few moments. For the people of you who don't know Jim, Jim is a brilliant neuroscience scientist. He got his PhD in the 90s from the University of British Columbia in Vancouver in neuroscience connected to psychology. He did an intensive molecular biology training in New York and became a professor in the Concordia University in neuroscience and psychology, where he served for a lot of years, and recently moved to do more research in Mexico, and he's jumping between Canada and Mexico sometimes. His main research with the animal concerning the neurochemical and molecular event concerning sexual behavior, and in human in objective measurements of desire and pleasure. You have more than 200 research papers in peer-reviewed journals, medical reviews, book chapters. He was the past president of the International Academy of Sex Research, but among others, is really fun to be with and to discuss and even to fight. He's a marvelous punk guitar player. He's really loving his animal. I know once that we talked with each other on the phone, and he told me that he had a car accident with his bicycle because he forgot something at the animal lab and ran back without thinking. And he loves his son very much when he spends a lot of time. So I will leave the stage for you, Jim. You will get the remote control and can move it as you wish. Okay. And for all of you, the audience at home, you have the Q&A to ask questions, and you can even chat with each other through the chat as well. And we will try to, at least I will forward the question to Jim if we will have time. Jim, go ahead.
Okay. Okay, I'm trying now to move the slide, and it is not moving. Ah, there we go. So, so stop it. All right, don't move. So I think there's a very long history of association of marijuana use with sex. And it goes back to, as you can see here, posters for the movie Reefer Madness, which came out in 1936. And, you know, you see the weed from the devil's garden that it produces reckless abandon, drug-crazed abandon, weird orgies, wild parties. And, you know, women cry for it, men die for it. I mean, these ridiculous associations. But the problem is, it was kind of true. There was a disinhibition that people experienced on marijuana, and why. So to understand where cannabis fits in our understanding of sexual behavior, you really need to go back to Master Johnson's EPOR model, which is really modeled after Albert Moll's 1908 four-stage model of the onset, the equable voluptuous sensations, the voluptuous acme at orgasm, and the sudden cessation of voluptuous sensations, which of course, thank God, Bill Masters put into excitement, plateau, orgasm, and resolution, because it was way easier to deal with. So you notice that and we all know this pattern. I don't have to tell you that men can have maybe one if they're lucky, two. Women can have three different patterns. Whether these patterns are, you know, really true, they were really based on a mix of EEG and cardiovascular activation, modified by Helen Singer Kaplan, who said, "What is excitement? What do you mean by excitement?" And she said excitement is a mix of desire and arousal, those two things that co-occur. And your very own Yannico Georgiadis added, sort of put it into the context of wanting, liking, and learning, the idea that you don't just have sex, but you learn from it, and you create patterns of behavior that occur because of that. If we think about the definitions of what we mean by that, sexual arousal has really been focused on increased genital blood flow, heart rate, sweating, pupil dilation, sympathetic and parasympathetic activation. But the genital activation is really, really critical. Sexual desire and interest, on the other hand, is sort of a more cognitive wanting or craving sexual activity, but it also can be looked at in terms of behaviors that are aimed at acquiring sex partners or sexual reward, for that matter. And what's reward? I mean, it's the pleasure you get from sex. It can be orgasm, but it doesn't have to be. It can be intimacy, bonding, control, other rewards. And of course, then what do we mean by refractoriness? We really mean sexual inhibition. That can be due to satiety. You've had enough, your brain is done. It can be due to primary aversion, secondary avoidance. If you're a woman with dyspareunia, the avoidance is there. In fact, the avoidance is there before you're going to engage in sexual activity. So if you imagine a sexual system that has hormones and circulating levels of those hormones activating both in the periphery and the brain, you've got sensory input coming from the genitals, you've got sensory input coming from the incentives in the environment. That's going to give you a net behavioral output, whatever it is, your interest in having sex, solicitation of sex, pursuit of a sex partner, and copulation. But interestingly, previous encounters that you have create an epigenetic change in your brain. Once you don't know what an orgasm is until you've had one. You don't know what an erection is until you've had one. That experience and expectation of what your sexual behavior is all about actually plays a role in activating hormones. So what you're really activating is arousability, the ability to be aroused, and attention. So attention placed on a sexual incentive, a potential partner, somebody who looks hot, whatever it is, that attention is placed on that so you can go after it. And that, of course, then if you mix that all with the sensory input coming from your own genitals and from the incentives in the environment, gives you that net behavioral output. Well, what drives those systems? Activation of these systems is produced by, especially of arousability, is produced things like norepinephrine, oxytocin. Activation of attention is much more of a motivational construct where we think of dopamine and the melanocortins that are driving hypothalamic dopamine. And when that system's turned on, you are turned on. When you're done, or when you're anxious or when you're stressed out, inhibitory systems come online. They come online again with reward and satiety, but also with stress and aversion. And these are the activation of opioids and serotonin. And what they will do is kind of, through their synaptic connections, will deactivate arousability by shutting off norepinephrine and oxytocin. They'll deactivate attention by shutting off your dopamine system, even though, you know, we know that opioids are going to sensitize dopamine and oxytocin, but that comes later. They're shutting it off locally to give you the refractory period that you might have. And of course, what that means, that shutdown is going to knock out this net behavioral output. So if you imagine this as a dual control system, something like the work of Eric Janssen, John Bancroft, Michael Perlman. Perlman envisioned it around a sexual tipping point that tips you if the physiological and organic issues, meaning the neurotransmitter systems, the hormones, are activated, and the psychosocial, cultural, and behavioral issues, well, I'm in the right place at the right time with the right person, that's going to now shunt you into excitation. And what you're really talking about is this kind of a system where drugs or states that activate those neurotransmitters, or drugs or states that inhibit the inhibitory neurotransmitters, are going to now push you into the excitement mode. When you're done or stressed out, drugs or states that activate these inhibitors or that deactivate the exciters are now going to shunt you into this refractoriness or into this mode of inhibition. Not tonight, I've got a headache. Not tonight, I've got a report due in the morning, whatever it might be. You're toggling between this, and you want a sexual tipping point that's relatively labile so that you can toggle into excitation, and if a bear comes into your tent while you're camping, you can stop being excited and get out. So when you look at the brain activation, as people like Yannicko has done, Georgiadis has done, you see that you're inactivating in red these excitatory regions of the brain, and you're also activating some inhibition over inhibition. So you get both excitation and disinhibition. Once you reach that voluptuous acme, as Moll called it, you're now activating inhibitory systems like frontal cortex, activated by serotonin, that's now shutting everything down. So you can actually see this toggling occur with brain activation in, say, an fMRI. So how do we get THC? How do we get marijuana and cannabis into this? Well, it's really going to become, or it is, a pharmacological aphrodisiac. The problem with understanding aphrodisiacs is that kind of anything can be an aphrodisiac. You have stimulants like amphetamines and cocaine, amyl nitrate, that activate you when you are sluggish. They activate sluggish excitation. Indeed, stimulants like the fills, sildenafil, tadalafil, vardenafil, etc., are going to activate a sluggish penis or activate a sluggish clitoris. That's all well and good, but then how might CNS depressants work? Alcohol, opium, even THC. I mean, as you know, Macbeth said, it provokes the desire but taketh away the performance. But there's going to be a dose at which performance can be activated as well. These are going to come online to inhibit inhibition. So the idea is if you're over-inhibited, now you're going to, or maybe anxious or whatever it might be, you're going to smoke pot and be less anxious and more able to focus. How might that work? Well, one thing to consider is that one person's aphrodisiac can be another one's dysfunction. The reason for that is the old inverted U-shaped curve that in this case is relating motivational strength to stimulus intensity. And that stimulus intensity can be both in terms of the intensity of what you're experiencing at the time, but it can also be your level of arousal, your general level of anxiety if you're in an anxious circumstance. And you can see that optimality occurs between this hypo and hyper arm of this inverted U-shaped curve, where excitation is going to drive you toward the hyper side and inhibition is going to drive you down toward the hypo side. But you can imagine that anything that might excite or stimulate autonomic outflow will work well for those with reduced outflow. But if you're hyper, it's going to reduce your motivational strength because now you have divided attention. And of course, likewise, states that inhibit the outflow may work well with those that are hyperfunctioning, so move you back toward optimality. But for those who are at optimal, it's going to knock you down. So if you're at optimal, you don't necessarily want to get so drunk that you can't have sex. And if you're at optimal, you don't necessarily want to get so excited that you can't focus. You need to remember THC is going to fit into this kind of a model, especially in a dose-dependent fashion. So what neural excitatory systems are there? I mentioned dopamine coming from both interhypothalamic and the medial preoptic area, but coming from the ventral tegmental area and the substantia nigra. This would be your motivational dopamine pathway, your mesolimbic pathway. This, of course, is your nigrostriatal or motor pathway. You also have noradrenaline, right? This is the reticular activating system. So your general activation, general arousal. And when you think about dopamine, one of the most important functions here from the zona incerta to the medial preoptic area is for this part of the hypothalamus to tell you what to do. And what it's going to do through its outputs is going to activate these systems that are your motivational and motor systems so that you can show appropriate motor action at the appropriate time for the appropriate incentive. Here's what the MPOA looks like in an MRI. You see it's activated just in response to visual erotica or visual sexual cues. These are non-clinical samples. In clinical samples, especially with hypoactive desire disorder, you don't get that activation. Here you see activation of the ventral striatum. Here you see activation of temporal lobe structures. The insula, for example, gets activated. This is very important because this visual erotica is kind of telling you and telling your body, okay, get prepared. And one of the main outputs from this medial preoptic area is its activation, or it's kind of co-activating in time sexual desire and your autonomic outflow so that you can actually have clitoral engorgement or vaginal blood flow, penile erection at the right time. It does so through a D1 receptor mediated event. Those D1 receptors are on GABA neurons that go to the ventral tegmental area and shut down these GABA interneurons. All right, they shut them down, which then releases dopamine from inhibition. What's interesting, note here, cannabinoid type 1 receptors are on the presynaptic terminals of those GABA interneurons, meaning that action here would have the same effect as action here to disinhibit. The inhibitory systems, driven by largely by opioids and serotonin, and of course interneurons in places that have those outflow, the opioid effects come online to induce reward, sexual reward, orgasm reward. That ecstasy you feel at orgasm is an opioid-mediated event. But the satiety you feel afterwards is a serotonergic mediated event. That's why it works well with antidepressant or as an antidepressant. By blocking reuptake, you become sated. Problem with that, of course, is when you are taking too much, you don't have any sexual desire or ability, or you can't have an orgasm because you're already there, at least pharmacologically speaking. And these serotonergic ascending pathways go to all the same places, but they're at war with dopamine. They end up activating GABA that shuts all the presynaptic dopamine down. So with dopamine activating your system, serotonin is now going to shut it down. And what's interesting is that serotonin in the cortex, especially prefrontal cortex, is controlling what's called executive function. And I'm sure all of you know what that is. You have forethought, you don't spend your savings, you problem solve, you can play the piano or play the guitar and play notes properly because you've got behavioral inhibition in a sequence. When there is a lot of serotonin in the activating the prefrontal cortex, you get behavioral inhibition. Behavioral inhibition is critical, again, to get you out of that tent when a bear is coming in, to get you to be able to stop what you're doing and do something else, to not spend your savings. But that inhibitory outflow from the prefrontal, which you see in blue, is going to places like the amygdala, like the motor areas of the frontal lobes, like the thalamus, like the medial preoptic area, and shutting down their actions, which means you're shutting down the excitatory systems. And of course, then that means that outflow from the preoptic area to the ventral tegmental area, which of course is activating dopamine going to the ventral striatum, isn't there. So you're knocking it down. If there's an incentive there, too much serotonin, too much inhibition in that prefrontal system is now going to say, well, I don't see that. I really need to attend to what I'm doing. Again, inhibitory outflow regulated by serotonin. So serotonin in the prefrontal cortex, activating a number of serotonin receptors, but 5-HT2As activate these glutamate pathways that activate another glutamate pathway that turns on that GABA inhibition in the ventral tegmental area. Now these have been determined. So if you were to knock serotonin out, you would knock out that, hence allowing this system to function. So again, excitation, inhibition, they're in a balance. Well, where do cannabinoids fit to get to the root of what we want to talk about today or tonight? And since antiquity, cannabinoids have been used, or marijuana has been smoked or eaten. Even there are cannabinoid actions or endocannabinoid in the brain actions induced by things like components in chocolate. It's been used as a sedative hypnotic. In fact, that's what THC is classified as an anxiolytic, as a hyperphagic to make you eat, as an analgesic, and even as an anti-inflammatory medication. Cannabis can be smoked, it can be eaten, it can even be macerated and put into a drink. So you mush up the leaves and you take an extract like a tea. Of course, in the 1920s and 30s, throughout the world, you had this notion that cannabis was a bad thing, that somehow doing this was going to, give you drug-crazed orgies and, put you in a position where your life would be ruined by you smoking cannabis. Here's Bob Marley, one of my great heroes, and I think Bob Marley made a very good point in thinking of cannabis's actions as being, in his case, religious, but in other people's cases, it was, treating their glaucoma, even though they could go to jail for this. It was treating, their anxiety, even though they could go to jail for it. So what do we know? We know that extracts of exogenous cannabis from the leaf have been used for millennia. There are medical texts from Mesopotamia and Egypt that are over 4,000 years old that describe extracts of cannabis being used for sexual, gynecological, and reproductive purposes. I've listed a number here. Used as an aphrodisiac, used to treat PMS or painful menstrual cramps, used to treat pain during sex, so things like dyspareunia or clitoridynia, used to treat menopause, not to treat the hormonal aspects of menopause, but to treat some of the way the body is now redistributing metabolism during menopause. And of course, even activating spermatogenesis and spermatozoa, activating the head of the spermatozoa so that it can actually penetrate the egg. Turns out that like the PDE5 inhibitors, sildenafil, other fils, local application to the genitals of THC actually stimulates nitric oxide, just as sildenafil does. And that relaxes smooth muscle, relaxes the blood vessels, allowing blood into the corpora cavernosa, and then stimulating the erection of the clitoris or the penis. Interestingly, local application does not get into the bloodstream. So people used to actually take macerated or cannabis indica fluid extracts, and they would rub it on their genitals. Now, people didn't know about the distribution of receptors then, but it turns out that cannabinoid type 1 receptors exist in the genitals, so we'll call it the little head, and exist in the big head. And so those actions become very important when you either smoke, eat, or drink or rub a lotion of cannabis on the body, because these regions of the body contain the receptors that are going to be activated by them. What are the cannabinoids? Well, Kobe told you about very many of them. There are very many of them. The ones that are biologically active, whether they're psychoactive or not, are derived from a precursor in the plant, right? Cannabidiolic acid, which then gets converted by different enzymatic degradation into different cannabinoids. So there's cannabidiol going through this pathway. Here's delta-9 tetrahydrocannabinol going through that pathway. This, of course, is THC, and this, of course, is CBD. the two that seem to, the balance of which seem to either have medicinal properties or psychoactive properties. In the liver, THC actually gets metabolized when you, not so much when you smoke it, but when you eat it, metabolized from this THC compound through a liver enzyme into 11-hydroxy-THC. 11-hydroxy-THC, and a big first-pass metabolism in the liver when you eat it especially, is an even more psychoactive compound. And it's going to get back in the bloodstream and give you even a greater high than you might get just by smoking. That, of course, is then activated by a different liver enzyme or metabolized by a different liver enzyme into the carboxyl form of THC. This is the inactive form, and then it can become a glucuronide conjugate. You find this inactive form stored in adipose tissue and secreted in urine and feces, which of course are the compounds that are checked in urine drug screening, especially in the United States, in the states in which it's not yet legal. Cannabinoid receptors exist kind of all over the brain, but in particular sites. For example, the caudate and putamen, which of course the ventral striatum is the bottom part of this. You've got the globus pallidus, you've got hippocampus and amygdala, you've got substantia nigra, you've got huge amounts of cannabis in the cortex of the cerebellum, and you've got moderate amounts in the outer layers of the cerebral cortex, okay? So, and you also find them in the hypothalamus, large amounts. So cannabis is acting in places, or these CB1 receptors are activating the brain in places where motivation meets physical activity, meets directed responding, also meets, here's the prefrontal, meets inhibition. CB1 receptor function. So you have agonists, meaning they bind to the receptor and activate, like the plant extract of THC, you have endogenous fatty acid molecules. So anandamide, which is an arachidonyl ethanolamide, or AEA, you have 2-arachidonyl glycerol, or 2-AG. These two are the endogenous or endocannabinoids. They're endogenous agonists. So your brain produces these naturally by the breakdown of different substances, but in this particular case, by the breakdown of diacylglycerol. You have a second messenger system called PIP2, or phosphatidylinositol bisphosphate. It gets broken down into 2 second messengers, inositol triphosphate and, oh, I don't know what that is, inositol triphosphate and diacylglycerol. The DAG becomes 2-AG, which can then either be metabolized into AEA or other substances. And these, it says my battery is running low, which I don't really understand. Hang on, give me 2 seconds. I am plugged in, so this should be okay. Okay, so let me just do this. All right, and plug it back in, and all right, good. So it gets broken down into these, which then bind to a cannabinoid receptor, and that is going to have an action to shut down calcium influx into that presynaptic neuron that's right next to it, which is going to shut down neurotransmitter release. You have antagonists like rimonabant, which are going to bind here but block the endogenous endocannabinoids, or block the exogenous THC, or CBD even, from activating that receptor and shutting down that neurotransmitter release. So this is a signaling pathway I just mentioned. I'm not going to go through that again. Well, there are dose-dependent and biphasic effects of cannabis on anxiety, and people have noticed this, just like there are on sexual activity, where low-dose THC, by kind of activating CB1 receptors in this CB1 serotonergic dimer, activate glutamate, which is sent from prefrontal cortex to the amygdala to shut down the GABAergic system there. And upon doing that, so instead of activating GABA, you're shutting it down. You're shutting down the system through disinhibition, GABA activates anxiety. You're now shutting that down to decrease anxiety. However, as the doses increase, you're now activating a different cannabinoid receptor. Kobe, I can't see it. What are you saying? Anyway, I can't see what Kobe's saying. You're activating a different cannabinoid receptor, which is now going to increase that glutamate and increase anxiety because you're increasing activation in this region. So what do we know? Well, there's level 2 to 5 evidence in females that acute low doses of cannabis increase sexual arousal and desire and the quality of orgasm in women. This hasn't been tested directly. That's why it's level 2 to 5 evidence. We know that high dosing to the point of sedative effects on motor function and increased anxiety can inhibit exactly what the low doses activate. So it's somewhat like what we see with anxiety. There are direct dose response studies and RCTs of THC and other cannabinoids that have not been done in humans. They've been done in animals, but not in humans. Peripheral THC, however, can increase vaginal and clitoral blood flow, which would then make you argue, though, okay, so low doses in the brain, local application to the genitals will activate. In rats, it's the same thing. Low doses of THC, because we can study this in rats without going to jail, will increase lordosis, increase solicitations, increase even ultrasonic vocalizations that are indicative of an orgasm-like response, or an OLR, in female rats that are primed with low doses of estradiol and progesterone. AEA, in fact, increases sexual motivation at a runway test for females. The inverse agonist, rimonabant, also increases lordosis, solicitations, and motivations, so probably counteracting a higher dose effect. High doses of THC inhibit solicitations and lordosis, and CB1Rs in the midbrain are downregulated during estrus. So hormones are actually setting up a system that's going to downregulate cannabinoid receptors in inhibitory regions, perhaps upregulate them in excitatory regions. In males, the story is somewhat similar, but somewhat different. Acute low doses of cannabis, whatever that means, because people are smoking it, increase sexual arousal, desire, and the quality of orgasm in men, and increase the duration of intercourse. Probably, as you increase the duration, you're increasing arousal, orgasm riding on higher arousal feels better, and that may be why men report this. Again, these are all subjective from memory reports. Chronic dosing can sensitize desire, but it also decreases erectile function, so it has an effect very much like alcohol. But notably, high chronic dosing decreases gonadotropin stimulation of testosterone synthesis. So in fact, high doses for a very long time will decrease testosterone synthesis. Direct dose response studies and RCTs, like in female and women, have not been done in humans yet. Now that Canada's legalized it, the Netherlands have had it legalized for a long time, maybe these studies could begin to actually be done. In male rats, acute or subchronic administration of CB1 agonists impair both intermissions and ejaculations. Acute administration of CB1 antagonists, like rimonabant, induce erections. They reduce the number of intermissions necessary for ejaculation and reduce the ejaculation latencies. Might be a good thing unless you consider that to be an animal model of premature ejaculation. So rimonabant also does this, and the high dose agonists, the high dose of THC also inhibit gonadotropin stimulation of testosterone synthesis. It's, again, very much like in humans. But AEA applied to the penis can stimulate erections, very much like THC applied to the genitals of females. So low-dose cannabinoid actions are going to, in the cortex, reduce behavioral inhibition because they're going to knock out that serotonin. They're going to do that here. They're also going to reduce anxiety in the amygdala, again, because you've knocked out that serotonergic activation. So you knock out the activation of foreboding. In the MPOA and VTA, endogenous cannabinoids, or THC, are going to block GABA synthesis in those interneurons, like I showed you before. The net behavioral effect of that is disinhibited dopamine transmission. So in essence, with cannabinoids, at least at low doses, you're deactivating inhibition and activating excitation. And the result of that then is increased arousal, attentiveness, desire, and enjoyment. Higher dose cannabinoid actions that are known, at least, there is an inhibition of oxytocin. And that may be, at least centrally, one of the reasons why you inhibit erection at high doses. So you get inhibited genital blood flow when these oxytocin neurons from the PVN are no longer secreting oxytocin into the bloodstream, right? So you get a decreased peripheral oxytocin, which then decreases the likelihood of erection. The other thing that high dose cannabinoid actions do, at least here in the basal ganglia, are going to produce the sedation and ataxia that you see with very high doses when you're sitting there listening to reggae music, but not being able to move. You also get anxiety with high dose effects in the amygdala, like I showed you before. Well, this is going to take away your attention from sexual cues, and this is going to make you unable to respond to sexual cues. The two things together, at least centrally, are going to knock out your ability to copulate itself. So again, think of this as a biphasic effect. If you have a dose response curve here, low doses are producing excitation, right, through the inhibition of descending inhibition. The higher doses are reestablishing inhibition. So you have inhibition of descending excitation. So you have inhibition of inhibition of descending inhibition, or at higher doses, inhibition of excitement. Okay. So the take-home messages for today's talk: THC exerts biphasic and dose-dependent effects on sexual arousal, desire, and orgasm intensity in women and men. They're biphasic because low doses facilitate through both disinhibitory and excitatory mechanisms, this is known, whereas high doses inhibit through the inhibition of excitation, augmentation of anxiety, activation of sedation, and motor ataxia. So I think when we can finally do RCTs with THC, we're going to see this kind of effect where as you increase the dose, you're going to start to get these inhibitory effects taking over. Evidence for both tolerance and sensitization occur. With some of these effects, baselines are critical, meaning you need to know where you are in that inverted U-shaped curve. If you're hypoarousal or hypoarousable, you don't want to be taking even a moderate dose. A little bit might go a long way. If you're hyper inhibitory, a lot may go a long way. THC applied to the genitalia increased genital blood flow locally with no central effect. This is important because people are beginning to put THC and CBD into ointments for genital use. That is, to rub them on the genitals as an ointment to increase the blood flow. THC absorbed through the lungs acts quickly with little first-pass metabolism in the liver, but THC eaten, meaning absorbed through the intestines, has a large first-pass metabolism in the liver and is converted to the 11-hydroxy form of THC, a more potent CB1 agonist. So the problem here is that if you smoke it and you eat it on that inverted U-shaped curve, you're going to be in different places because of the greater psychogenic activation of the 11-hydroxy THC. Chronic high-dose THC reduces gonadotrophin release in men, leading to low circulating testosterone, low sperm count, and impeded sperm efficacy. We don't exactly know what it does in women yet, but you can imagine if release of testosterone at ovulation is one of the important components of desire, if you're doing this chronically, you may in fact get a reduced release of androgens. So again, more and more countries, Canada, for example, have joined the Netherlands in legalizing cannabis possession. There are other countries like the US have many states that have legalized recreational use. Well, I have to say, given the dearth of controlled studies in humans, the lack of controlled studies in humans, perhaps the time is right for sex researchers in Canada and in the Netherlands taking the lead to seize the initiative and start doing some real studies on THC and see whether THC or other cannabinoids can actually be used to enhance pleasure, enhance desire, and knock out anxiety. I think that's critical. And with that, I will thank you for your attention, or say dank you and nog vragen.
Thank you so much, Jim. Really amazing, a lot of information. And if one of the audience have a question, please let us know. In the meantime, we have a few minutes also for questions. So I have one question from the audience, and the question said, the women in menopause complain of bad sleep and start taking cannabis. Should we discourage them? What is your opinion about it?
Well, I mean, you know, one of the things that cannabis can do when sleep problems occur is it can induce that sedation, which will help you go to sleep. It can reduce anxiety, which can help you go to sleep. The problem is if you go off it, you're going to have this, many people that do this have kind of a rebound effect where now they can't sleep or they have disrupted sleep. Again, the hypothalamus is a very important structure for, circadian rhythms and whatnot. Sleep problems that occur in menopause are largely targeted from those regions in the hypothalamus and part of that whole circadian thing. So your sleep cycle goes off, you become phase advanced, and some women become phase advanced, some become phase delayed. They complain of not sleeping, but in fact, they're going to sleep on the couch at 6 p.m. and waking up at 11 going, oh my God, I forgot to have dinner. And there's many things that are happening to that system. Cannabis might be able to restore that, but if you go on it long enough and you try to go off it, you'll have the same effects that you get with sleeping pills, which are, GABAergics, where you're now going to get a rebound effect. So I can't say whether you should or shouldn't. We have medical cannabis, but we don't have RCTs that tell us exactly how much you should smoke. Again, this is why I say know your baseline. It's kind of critical.
Thank you. In the meantime, we've got another few questions. Is there any experience in the treatment of genital pain syndrome, mainly in the painful ejaculation, for example?
Some people who have that use cannabis and they find it goes away, but probably there are others who hear about that and use it and it doesn't go away. And this is why we need, again, good clinical trials of this to figure out, is there a dose dependence of it? Is there, if you smoke it, it doesn't work, but if you use it as an ointment on the genitals, it does. These are going to, we have our work cut out for us for the next decade, I think.
Okay. And another question, the strong dose dependent effect, are with use in clinical work?
Obviously, I don't think anyone has actually, in a responsible way, said, yes, you need to smoke 2 joints and that'll be it, because we don't know. And the quality is going to be different, right? So you may go to one coffee shop and, this cannabis will do you, but in that other coffee shop, the same spliff doesn't do you. Why would that be? I mean, they could be coming from 3 different plants, as Kobe, as you mentioned, but it's also the case that even from the same plant, and generated by one cannabis provider may have THC that's 20%. The other plant the same plant, but coming from a different supplier, may have THC that's 10% or 12%. So, how much do you drink of liquor as opposed to wine, right? Because the wine may be a lower percentage alcohol, so you can actually drink more before you get drunk, but the drink may put you there because the higher percentage. We need to know exactly what the percentages are and work off of that. So we don't know that yet. So if I were a clinician, I would not be saying, oh, well, do 2 or do 3 or, maybe find out for yourself, maybe if you know. But there's no responsible clinician who's going to say, do this amount, because we don't know yet.
You probably saw in the beginning, in the Netherlands, we have already standardization. Some pharmacists have a standardization of the amount of THC and CBD, that, as I show in the diagram there. But my main problem is that you talk a lot about the low dose and the high dose. I couldn't find nowhere a reference of what is low dose and what is high dose.
Exactly.
Maybe you are more aware of it.
We can do this in animals because we can just give them THC, right? But you don't know what a low dose is in a human yet, right? I mean, is a low dose half a marijuana cigarette? Is a low dose, 1 brownie is a low? We don't know yet. And a low dose is going to be an actual dose, right? Either that you're smoking or that you're eating. It's not going to be 1 marijuana cigarette or 1 spliff or 1 brownie. You know what I mean? And it's going to be different for different people.
Yeah, that's why standardization is a key issue in that respect.
Exactly.
And the standardization is also because actually we try not encouraging smoking because even that effect is not known, and the same go for for eating because the pharmacokinetic of eating in different species is also not very known. So actually, the aerosol is more preferred, I will say, and in the standardized medical cannabis, you can know the amount and compare it more or less. But
that's right.
Then we face the fact that we don't really know how much the dose, because the information that we have is in the few studies in human are very limited concerning the doses.
Yep.
And that's also pointing to another two question that we have about the specific stimulant and relaxant is not about the type that you can buy in the coffee shop, because there is not standardized and you don't really know. But if you want to use it with your patient, at least you should know what is the percentage of THC or CBD in that respect. And even what mentioned as pure CBD sometimes is not pure CBD.
Yes.
Another question, Jim, is concerning, you mentioned the testosterone synthesis, and we know it for the male, but you also know that androgen is also needed for the female. So the question is, do we have information about it, the effect of cannabis on androgen in female? And do you have information about the effect of estrogen or progesterone?
Sorry, say that again.
You already answered that about the androgen in female, we don't have any information, but do we have an information about the estrogens?
No, we don't. I mean, the only thing we have is one finding that showed that CB1 receptors, that estradiol reduces that. So what's the effect of CB1 agonism on estradiol levels? Hasn't been done yet in rats or humans.
So we have a lot of work to do, actually.
That's it.
Okay. If there is any other question, I'm sure that Jim will answer.
No, there are some. Somebody asked, are there known drawback term chronic local topical application to the genitals? Not long term, and in fact the topical application is just now beginning to come out on the market. So again, more work to do to try to figure out if that's true. The good thing about the topical application is it doesn't get into the bloodstream, so it works locally. All right, so that's an amazing, wonderful finding, because if you're able to put it topically and get increased blood flow, you don't have to worry about the central effects. And so it may not be a dose for dose thing. You can actually have a clinically effective dose that you could apply to the genitals, but we don't know what long-term effects there might be.
I heard about a company who is interested in producing a lubricant with THC.
Yeah, right. And of course, the reason why it would work is because it's activating genital blood flow, and hence activating, mucus secretion in the vaginal mucosa.
Yeah, so that will be interesting, but we'll need much more research and good randomized trial to know. But actually, we need to go to the basic and start with the pharmacokinetic and pharmacodynamic of the THC in the different doses.
No, that's absolutely right.
So
I mean, it's unfortunate. We've had. It's just unfortunate that the drug was made illegal, in order to target particular populations of people in the United States. It was and then it just became this thing the world over. And, 100 years of people going to prison who are using it for, to treat their glaucoma, to treat chronic pain, to treat PTSD, to treat themselves. They didn't have that, so they went to alcohol, or they went to some other self-medication. And now we're finding all of these amazing things that different cannabinoids do and that THC does. And it's like, wow, 100 years of, closing your eyes to that when we could have had something that could be medically beautiful. It's sad.
Another interesting question, Jim. You see it about the pathway, gendered?
Oh, is the cannabinoid pathway gendered? That we don't really know. People have looked at CB1 receptor densities in the brain of rats, and we don't see a male-female difference there. Whether the pathway is gendered or engendered differently in males and females is something that we don't know for humans yet. What we do know, though, is that estradiol seems to downregulate CB1 receptors, and in males, testosterone converted to estradiol in the brain downregulates CB1 receptors, at least in the midbrain. So it doesn't seem to be gendered that way in terms of functionality, but whether progesterone interacts with it in a way that men will never understand is unknown, right? We don't really make that, so it's not a bisexual hormone, shall we say. It's a non-gendered hormone. It could very well be that in states of progestation, cannabinoid receptors are doing different things, and they may have reproductive function that way. We don't know that either.
And the last question that I see is, are the effect on the rectal function temporary during the use or permanent? And it's a clear temporary. And actually, if you think about the availability of THC on the bloodstream, normally it's reduced in 2 hours.
That's
and
right.
This is not going to the bloodstream, so I expect that the effect will stay for 1 to 2 hours at most.
Yeah, I think that one of the things you see when people are going off of cannabis, chronic smokers, for example, the erectile function, which may have been reduced for a time, actually springs back. And it doesn't seem to matter how long you've been doing this, if you've been a chronic smoker for many years as opposed to just a few weeks. Oh, I see one more question. Is gabapentin a better option?
Gabapentin is related to the sleep issue.
Yeah. I mean, gabapentin is good for sleep, and you get less of a rebound effect with gabapentin. So people that have used it, that is relative to people that are taking cannabis chronically, who then experience sleep problems getting off it. There's less of a rebound effect with gabapentin. So at least for sleep problems, it might be a better option than cannabis.
But there is a lot of side effects.
Exactly. Well, that was the next thing I was going to say. But yes, there are side effects that would, again, things are in a balance, and you can do something and say, okay, I need to sleep tonight. I'm going to do this. And then tomorrow night you can't sleep because you need to go off it. And so, I'll do it one more time. And, weeks later, you've been taking it every night to get to sleep, and you're waking up groggy and unable to function.
Yeah, exactly. Another question, Jim?
Yeah, the question is, one joint a day keeps the doctor away. Well, I mean, for some things that might be true. And, it's not even a moral issue at this point. I mean, people are using it and have been using it to self-medicate. And, there are, if you're disinhibited, if you're an anxious person and you smoke one joint and you're not anxious anymore and you can function and be in the moment sexually, or be in the moment without pain, or be in the moment without your anxiety, you know what? Why not?
I know. But actually, from what I understand from your lecture, is dose dependent. So stay low and don't go high, and don't do it chronically. Actually, you may have the benefit on inhibition of the inhibition.
Right.
Am I right? And
absolutely.
And that's the key message. And that we need to have a lot of studies to understand it very well, from basic to clinical as well. Oh,
we have
the last question.
In combination with an apple. Well, I wonder what Eve actually really did offer Adam.
Yeah, we never know. Thank you very much, Jim. It was a very nice experience of a webinar, and I would also like to thank the audience at home, and also the ones who ask questions that are very interesting and as well. I hope all of you enjoyed from it as much as I enjoy it, and let's hope that NVVS will arrange more of this kind of webinars for everyone. And
I think so.
Excellent.
And Taku.
Yeah.
Taku.
Thank you very much, Jim.
Dank u wel.
Thanks, Jim. It was a very interesting talk, and we will be organizing more webinars in the future.
Excellent.
Okay. Have a nice evening, everyone. Sleep well.
Sleep well and have a beautiful evening. Bye-bye.
Dank u wel.